In this lecture, we'll explore the visual processing pathway corresponding to the transfer of neural information from the retina to the cortex. In part 2, we'll refamiliarize ourselves with the receptive field properties of retinal ganglion cells. Our next stop along the visual processing pathway is the lateral geniculate nucleus, or LGN. This is often referred to as the relay station between the eyes and the cortex. In part 4, we'll arrive at the first stage of cortical visual processing, known as the primary visual cortex, or area V1. It's here that we observe some remarkable changes in receptive field properties. In part 5, we'll further ascend the processing hierarchy and take note of how the brain combines information across space and time to generate percepts of whole objects. Part 1. The visual pathway. When we look at the visual environment, light from the distal scene is optically inverted. Consequently, light located in one's left visual field is projected onto the right-hand retinal surface in each eye. Conversely, light from the right visual field is projected onto the left retinal surface in each eye. This optical inversion, or flipping of the image, is known as a contralateral representation of visual space. This contralateral representation is actually preserved throughout the visual system, all the way up to the back of the brain, to an area known as the primary visual cortex. Consequently, the left primary visual cortex represents the right half of visual space, and the right primary visual cortex represents the left half of visual space. Neural information is transmitted from the eyes via a bundle of fibres known collectively as the optic nerve. Most of this retinal information is transmitted to a structure in the thalamus known as the lateral geniculate nucleus, or LGN. Information from the LGN is transmitted all the way to the back of the brain to an area known as the primary visual cortex, or area V1. A remarkable feature of the primary visual cortex is that neurons, which are adjacent to one another in the brain, tend to represent adjacent regions of visual space. That is to say, there exists a map of visual space, albeit a distorted one, on the surface of the cortex. This is known as retinotopic organization. Part 2. Retinal ganglion cells. As we saw in our previous lecture, the final stage of retinal processing involves the ganglion cells. These are the cells which decide whether a neural response to a given image will be transmitted to the brain or not. How do they decide? That is determined by their receptive field structure. The black and white discs that you see in this slide represent a ganglion cell's receptive field. The white vertical lines at the bottom of the screen represent the cell's electrical activity across time. When the cell experiences net excitation, there's an increase in electrical activity across time, and when it's inhibited, there's a reduction in electrical activity. So, what stimulates this ganglion cell? Because its receptive field has an on-center and an off-surround, a spot or a strip of light falling across its central region will stimulate or excite this cell. If, and only if, the inhibitory surrounding region isn't also strongly stimulated. In this slide, we see that the bar of light covers the receptive field's central excitatory region entirely, but only partially covers its inhibitory surround. So, we observe net excitation. In other words, the ganglion cell will fire. In this slide, however, the bar of light stimulates only an inhibitory region of the cell's receptive field. So, overall, the cell will be inhibited. An important feature of these ganglion cell receptive fields is that they're circular. A consequence of this is that they can't differentiate between bars or strips of light of different orientations. The only thing that these receptive fields are sensitive to are differences in light which occur across their centre and their surround. Part 3. The lateral geniculate nucleus. Once a ganglion cell is stimulated, its electrical activity is transmitted through the back of the eye via the optic nerve to the lateral geniculate nucleus, or LGN, which is located in the thalamus. The LGN is organised in discrete layers, which can be seen in the image on the left. The deepest layers of the LGN, shown here in red, and which are referred to as the magnocellular layers, are comprised of neurons with relatively large cell bodies. The upper layers, shown in blue, are called parvocellular layers by virtue of their relatively small cell bodies. The different layers each receive information from a specific eye. There are functional differences between the magno and parvocellular layers. The magnocellular layers are composed of neurons with large receptive fields, and so these respond to coarse, low-resolution spatial information. By contrast, the parvocellular layers are composed of neurons with small receptive fields, and so respond to fine, high-resolution image details. Another functional difference is that the magno layers respond to rapid changes in the image, such as occur when an object moves. By contrast, the parvo layers are relatively insensitive to movement. Finally, whereas the parvo layers contain neurons that are sensitive to colour, magno neurons are insensitive to colour variation. Part 4. The primary visual cortex. As we mentioned earlier, the neurons in the primary visual cortex are retinotopically organised. Neurons which receive information from one region of retina tend to be adjacent to neurons which receive information from an adjacent region of retina. Accompanying this transmission of information from the LGN to the cortex are some truly remarkable transformations in receptive field structure. Whereas retinal ganglion cell and LGN receptive fields are circular, the receptive fields of many neurons in the primary visual cortex possess a strong orientation preference. For example, a V1, or primary visual cortical neuron, may be strongly stimulated by a vertical bar. However, as the orientation of the bar changes, the neuron may respond less vigorously, and eventually it might fail to respond at all once a critical orientation difference is reached. This discovery of orientation selectivity in primary visual cortical neurons earned those who discovered it, David Hubel and Torsten Wiesel, the 1980 Nobel Prize for Scientific Research. This short film shows the mapping of receptive fields in primary visual cortex. As you'll see, the neuron whose electrical activity is being measured shows a strong preference for certain orientations. It must be emphasized that sadly, many thousands of animals have been sacrificed in the pursuit of this fascinating and foundational work. Other technologies, such as functional magnetic resonance imaging, have become sufficiently advanced so as to enable researchers to safely demonstrate the existence of orientation selective neurons in the primary visual cortices of human subjects. All of this begs the question, why does the brain possess orientation selective neurons? What functional benefit or information does orientation selectivity confer? A simple answer is edges. An object's edges are a powerful cue to segment and to identify objects. A compelling demonstration of the importance of edges to object recognition is in the line drawings produced by humans the world over. To represent an object, a child's first instinct is to represent the object's outline structures. Yet another demonstration of the significance of orientation-specific contours for the human visual system is in systems of writing. These letters, or characters, are each composed of simple lines and curves. Part 5. The cortical visual processing hierarchy. The orientation-selective neurons which occur in primary visual cortex are only a preliminary stage in object recognition. Once a neuron in V1 is stimulated, this information can then be transmitted to other visually sensitive neurons elsewhere in the cortex. The brain areas associated with object recognition are shown as coloured regions in the image on the left, the so-called ventral stream. As the information is transmitted from one region to another, the receptive fields become both larger and more complex. Whereas receptive fields in V1 are very small, meaning they respond to a small region of the visual field, higher level neurons in, say, area V2 are roughly twice the size. In addition to their size, the type of information that these higher level neurons respond to becomes ever more specialized.